today. And it's now my honor to introduce today's keynote speaker. And that keynote speaker is Dr. David Fajgenbaum. And some of you will know some of David's story about how he went from a healthy athlete to a critically ill patient virtually overnight. And his illness stumped expert after expert who could not figure out how to treat his condition. Well, David, he's a force of nature, as you will get a taste of here during his keynote address. He sort of took control, and he became a leading expert on Castleman's disease, which is the disease he's afflicted with, and generated new sites regarding the disease and new insights about how to find effective treatments. And today, Dr. Fajgenbaum is an assistant professor of medicine at University of Pennsylvania. He's the associate director of the Orphan Disease Center. He's director of the Center for Cytokine Storm Treatment. He's the co-founder of the Castleman Disease Collaborative Network. And he's the leader of 18 clinical research studies, including a natural history registry of Castleman's disease. So I think it's clear that David is the ideal keynote speaker for our symposium today. David, over to you.
Bill, thank you so much for that introduction. What an honor it is for me to be a part of this really important meeting and to be able to share a bit with all of you about my journey. I'm going to begin by sharing my screen. So, Bill, my talk today, framed in the context of this really important meeting, is about cancer registries and personalized medicine. Personalized medicine is a term that we hear a lot in medicine. I'm going to be sharing my own take on personalized medicine. So today I'm going to share about really 5 perspectives that I think are critical when we think about taking down cancer and really generating the data to do that. The first is that of a caregiver. This is a picture of my mom and I shortly after her diagnosis with brain cancer. Watching her battle and watching the way her doctors took care of her inspired me to want to go into medicine shortly after she passed away from battling glioblastoma. A few years later, I was a third-year medical student at the University of Pennsylvania, hoping to become an oncologist and treat patients like my mom, when just as Bill said, I became this critically ill patient, seemingly out of nowhere, with multi-organ failure, and even had my last rites read to me when my doctors didn't think I would survive. But it was realizing that I didn't just need to survive, I needed to actually get involved in research if I wanted to live long term. And so I started searching for treatments that could maybe help me. And then I realized that it wasn't something I could do on my own. It was something that I would need to become an advocate, to connect with other physicians, researchers, and patients to really make progress. So I'll be sharing these 5 perspectives today as we think about rare cancer registries. This is just a schematic that I wanted to highlight how in order to make progress, we really need the patient perspective, the physician perspective, advocates, we need researchers, and we need loved ones of patients, caregivers, to be a part of the conversation, to work with FDA and with pharmaceutical companies so that we can really make a difference for individual patients. And also that we can think beyond an individual patient towards the entire disease field. And what I'm a huge proponent of is actually learning from one disease field and applying it to others. So thinking about how do you apply this network across diseases and things that we learned for, say, Castleman disease to rare cancers. So today I'm going to begin by sharing my personal journey, share about the collaborative natural history registry that I run for Castleman disease, how it's helped us to lead to novel therapeutics for Castleman disease, share about some applications beyond, and then finally close with some lessons learned. So as I shared before, I was a healthy third-year medical student, hoping to one day become an oncologist at Penn. And then out of nowhere, I started experiencing multi-organ failure. My liver, my kidneys, my bone marrow, my heart, and my lungs all began to shut down. And I was hospitalized due to a cytokine storm. My immune system was literally attacking my vital organs, but we didn't know why. We had no idea what was causing it. Over the course of 11 weeks, I gained 70 pounds of fluid. I drifted in and out of consciousness. I even had a retinal hemorrhage that made me blind in my left eye. All of this without a single diagnosis. But thankfully, the diagnosis was eventually made within about 48 hours of having my last rites read to me. because the doctors didn't think I would make it. And with the diagnosis of idiopathic multicentric Castleman disease, my doctors were able to begin treatment. IMCD is a rare immune hyperactivation disorder. We actually don't know whether it's caused by somatic mutation, where we would call it truly a cancer, a lymphoma, or if it should more appropriately be considered an autoimmune condition. It really sits at the intersection of autoimmunity and oncology. But because the immune hyperactivation is so deadly and we don't know how to stop it, I was given multi-agent chemotherapy, a combination of 7 different chemotherapies that completely obliterated my immune system, but actually started helping me to feel better. I felt better with every dose of Adriamycin, Cytoxan, and Etoposide because I was so sick from my immune system hyperactivation. And over the course of, this is almost 5 months into my disease course, you can see my dad and I sitting up. I'm just so thankful that the chemotherapy has kicked in and is saving my life. This is another picture of my dad and I a few days later. This actually happens to be New Year's Eve of 2010. And a few minutes after this picture was taken, we were walking around the hematology oncology unit, and we passed the family waiting room, and we noticed that there was a gentleman who was kind of swaying in his chair. He was clearly drunk on New Year's Eve. And on our next lap, my dad saw that this guy had fallen onto the ground, and so he helped him back into his chair. And he looked at my dad and I, and he said, "Thanks so much. Good luck to you and your wife." We were really confused at first, like, wife, what is he talking about? And then I looked at my belly and I realized that he thought that I was my dad's pregnant wife, which was a low point emotionally. But we laughed really, really hard. And maybe you can see how someone maybe thought that I was my dad's pregnant wife with the big belly from all the liver and kidney failure I was experiencing. About 7 weeks after that moment, I was finally discharged from the hospital. And you can see that I've still got a big belly from the liver and kidney failure. I lost all my hair at this stage. And this is a picture of me from just a few years before. I played college football at Georgetown. And I always say this is the worst before and after picture of all time. But if we could flip the order, it would be this amazing advertisement for Peloton or for Muscle Milk. But unfortunately, the pictures are in the wrong order. So about 6 months later, I was able to return to medical school. This time I was on an experimental drug, a drug called siltuximab, that blocks interleukin-6, one particular cytokine in the cytokine storm. And we were really hopeful it was going to keep me in remission. So when I returned to Penn, I got involved in the new orphan disease center that had been started at Penn. But unfortunately, about a year after getting out of the hospital, I had a full-blown relapse, this time on the only drug in development for my condition. And my doctor explained to me that there were no more drugs in development and that I had literally run out of options. And so I turned to my dad, my sisters, and my girlfriend, and I promised them I would dedicate the rest of my life, however long that may be, to trying to identify a treatment, maybe even a cure for my condition. So the first thing I did was find a lab on campus where I could get a little bit of space and start conducting laboratory research. I could get access to my own blood samples. I couldn't get access to too many other samples at the time, but thankfully a colleague provided me some space in her lab. And I started a foundation called the Castleman Disease Collaborative Network because I knew I couldn't make the progress we needed on my own. I knew that I needed to bring together physicians, researchers, and patients to advance research, and I knew that we needed to take a different approach. There were already 2 foundations in the Castleman space that were doing research the traditional way, raising money and then putting out requests for proposals. We wanted to change that, and we came up with a new approach we call the collaborative network approach, where physicians, researchers, and patients crowdsource and provide ideas towards research, and then we prioritize those ideas and then go out and recruit the best people in the world to do the work. And this has really just been a game changer for our community. But there's an important part to that message of prioritizing and then seeking the right people to do that. And that's that we couldn't just wait and hope for the right research to happen. And this is a quote from my book, From Chasing My Cure. Hope cannot be a passive concept. It's a choice and a force. Hoping for something takes more than casting out a wish to the universe and waiting for it to occur. Hope should inspire action. And when it does inspire action in medicine and science, that hope can become a reality beyond your wildest dreams. For me, this was the moment in my life where everything changed, where I went from hoping that some researcher somewhere would figure out a treatment that could save my life to saying, if I'm going to hope for it, I need to spend my life and do everything I can to try to find that treatment rather than hoping for someone else to do it. So when I got out of the hospital, I tried to get a sense for what's going on in the idiopathic multicentric Castleman disease space. And I found that there was about $10,000 a year being invested into IMCD research, which, as you all know, is too little to make any sort of meaningful progress. The NIH had never given any funding towards IMCD research. There were no patients in a registry because no registry existed. There were two advocacy organizations I mentioned, but there was no sort of research agenda or centralized research plan. There were no cell lines, animal models, biobanks. The disease, as you can imagine, was very poorly understood. There were no diagnostic criteria, no treatment guidelines, no blood tests you could run to predict who's going to get better on chemotherapy or who's going to get better on IL-6 blockade, no FDA-approved treatments. But thankfully, there was that one drug in development, siltuximab. The drug that I mentioned was tried on me under emergency compassionate use from the FDA, but unfortunately, it did not work for me. So a little bit about IMCD and kind of what we were up against. So I mentioned IMCD is a cytokine storm disorder where there's incredible immune hyperactivation, but we don't know why that happens. We don't know if there are autoimmune mechanisms that are leading to the immune hyperactivation. We don't know if there are neoplastic cells, and this really is a lymphoma, or maybe there's some pathogen that we just haven't discovered yet that's igniting the cytokine storm. But thankfully, we did learn over 20 years ago that interleukin-6 was critical to the cytokine storm because Kazuo Yoshizaki, who's photographed here, discovered that interleukin-6 was elevated in a number of IMCD patients in Japan. Based on that observation, he wanted to develop a drug, which many of you all will be familiar with, called tocilizumab, that blocks the receptor for IL-6 with the intent of treating idiopathic multicentric calcium disease patients in Japan. And I'd heard a rumor that before he tested it on patients, that he tested it on himself. And so when I asked Kazuo, I said, Kazuo, I heard that you gave yourself tocilizumab to prove that it was safe. And he said, no, I didn't give it to myself. The nurse, she gave it to me. So Kazuo Yoshizaki made the discovery that IL-6 was a target. He developed the drug. He gave it to himself to prove that it was safe. And then he went on to give it to other humans in a phase 1 trial in Japan, which led to the approval of tocilizumab in Japan for idiopathic multicentric calcium disease. And of course, you all will be familiar that tocilizumab or Actemra came to the United States and was eventually approved for rheumatoid arthritis and also for CART therapy, the cytokine storm that occurs with it. That work inspired work by Janssen Pharmaceuticals to develop a drug called siltuximab, but rather than blocking the receptor for interleukin-6, it actually blocks the ligand directly. But we know from research that if you give a patient tocilizumab or siltuximab, that about 1/3 of patients will benefit from IL-6 blockade. Unfortunately, for the 2/3 of patients who don't benefit from IL-6 blockade, patients like myself, we don't know what else to do other than using cytotoxic chemotherapy. So my research focus really became, let's tease out what's happening biologically in the IL-6 refractory patients so we can identify drugs other than chemotherapy. Many of you all will know that this drug that Kazuo developed was used to save the lives and has been used to save the lives of thousands of CART therapy patients who got cytokine storms after their CART therapy. This is a picture of Emily Whitehead, the very first pediatric patient to ever get CART therapy, who was dying in the ICU just next door to me at the Children's Hospital of Philadelphia before tocilizumab saved her life. This drug developed for Castleman disease many years ago, saving Emily's life and likely also preserving the CART therapy program. I suspect that if young Emily had passed away, that it really could have led to in addition to incredible heartache for her family, really slowed down the entire cellular immunotherapy program worldwide. Many of you all may be familiar with the use of tocilizumab off-label for COVID. The cytokine storm that we see in idiopathic multicentric calcium disease is actually very similar to the cytokine storm that occurs in COVID-19. And as a result, many individuals have used tocilizumab off-label. And it actually got a lot of interest early on. And I think many of us are very hopeful that tocilizumab was going to be the solution for the most sick patients. And actually, a number of trials suggested that. But as time has gone on, and larger studies have been done, phase 3 studies, it's less clear whether it's helpful. There was a very clear negative result, and then more recently, just in the last couple of weeks, there was another result suggesting benefit. So the jury's still out. But as we shifted gears towards understanding what could we do to better understand treatments and therapeutic options for Castleman disease, I again knew that I couldn't do it alone. And so I was a medical student at the time. I enlisted a number of my classmates to join our team. And then I was a business school student and enlisted Wharton students to help me to build this team and to take on Castleman disease. In this picture, you see we're doing what we call the Castleman Flex. The disease is named after Benjamin Castleman, but we've kind of repurposed it as a warrior. And if we were all here, if we're all together today, I would have asked all of you to do a warrior flex, but I hope you'll do a warrior flex by your computer. I also knew that we need to engage the entire global physician and researcher community, and we've done that. And really, it's through connecting with these physicians, scientists, and patients all over the world that we've been able to lay out the right research agenda for progress. This is one way I like to think about drug development, particularly drug repurposing. It's around identification and validation of drug candidates and therapeutic targets. There are a number of ways to do that. It's about really harnessing and leveraging real-world data, whether it's off-label drug use, or well, it's always off-label, but intentional off-label drug use, or maybe observational studies where individuals are exposed to a drug that's not for the primary indication. Of course, moving forward to interventional studies, and then working towards FDA approval. Of course, if it's a not yet approved drug, it requires FDA approval. But if it's a drug that's already approved for something else, there are a number of conditions where we're doing really high quality trials and it can really change clinical practice without actually having a label change. But there is one part of this that I feel is essential, and that's why I believe I was asked to speak today, and that's that it is so critical to utilize the data that exists to establish the natural history of disease and to track treatments that are used for that condition. We heard earlier about how natural history studies and registries can actually help to inform endpoints for clinical trials. That is absolutely critical. And in addition, what was also mentioned is that registries and natural history studies are often used as post-approval measures to actually phase 4 studies after approval to track the use of these drugs. And so I'll be sharing about our experience with Castleman disease that's actually meeting both of these objectives. So turning over towards our Castleman disease space. So a few years ago, we decided we wanted to launch a collaborative natural history registry for Castleman disease, and there were a few reasons for that. The first is that the EMA required Janssen Pharmaceuticals, which was the makers of that IL-6 blocker siltuximab, to launch a registry back in 2014. It was not required by the FDA, but EMA did require it. In parallel, about 2 years before, the CDC and Scientific Advisory Board prioritized launching a registry as a priority for the field. And so the CDC and UPenn and Janssen worked together to come up with this tripartite agreement where we would both meet the EMA requirement that they had imposed on Janssen, while at the same time harnessing incredible amounts of data to better understand the natural history of Castleman disease and therapeutics. And so just kind of boiling it down, we wanted to do this because this is a rare disease with a small patient population, so limited data previously available. It was poorly understood, and there was a lot of off-label drug use that we wanted to get a sense for, in addition to meeting the CMA requirement. So the first thing we did was try to get a sense for, well, how do these registries typically collect data? What's the typical registry approach, data collection approach? You're likely all familiar with the kind of classic patient registry where patients go online, they enroll in high numbers because you can access patients quite easily online. Because it's patient reported and because the medical record is not available often, there's a small amount of data that you can definitively get from a patient through a traditional patient registry. You can't get every laboratory test ever run, every pathology report on a patient. You can get patient-reported outcomes that are critical, but you end up getting a smaller amount, a quantity of data than you would from the medical record, but it's low cost. The next approach is what we consider physician-directed, and this is the traditional natural history study, where you select 10 sites and those 10 doctors enroll all the patients they see with that given disease. So you have a low number of patients because if it's a rare disease, you'd need to have 100 sites or 1,000 sites to really enroll high numbers. So you're going to have a smaller patient enrollment, limited scope, but you're going to get increased quantity of data. The physician is going to have access to laboratory data, imaging data. There's a lot more data that can be entered. And of course, the cost is higher. You have to pay the physicians and the sites for their time. The third approach involves manual EHR extraction, where you actually manually pull data from the electronic medical record. You're going to have low patient enrollment because you have to basically reinvent the wheel for every single EHR. You can do this at Penn, and then you can do it at Sloan Kettering, and then you can do it at each institution, on and on. But the numbers are going to be low because it's an institutional focused approach. The quantity of data is going to be huge. It's from the EHR, and the cost is expensive because it's a system-by-system approach. The fourth approach would be insurance claims data, leveraging ICD codes to find patients. And so now you're going to have a huge number of patients at your disposal. The quantity of data is kind of in between. There's, in some cases, you get a lot of data on use of medical resources. You don't necessarily get laboratory data and insights that'll come from the EHR, and it's pretty expensive. And so when we looked at all these options, we said, well, let's come up with a hybrid, what we call a natural history registry, where we combine the best of the patient-reported piece with the best of the physician-directed piece. And I'll go through that now. We call this approach, and we call it the Accelerate Natural History Registry. And we've been doing this for the last 4 years, and I'm really, really pleased to share the way that we do it. So it's a hybrid approach in that patients enroll online, they consent online. As soon as they enroll, they provide patient-reported outcomes, and they continue to provide PROs every 3 months. But once they enroll, they give us permission to access their complete medical records. So we reach out, we get the complete medical records sent to the University of Pennsylvania. At the same time, we also request any biospecimens, lymph node tissue, bone marrow, any samples that are left over or that are available. Then we have manual data extractors. We have 3 full-time employees who go line by line through every single medical record and extract out thousands of data points per patient. That data is then shared with an expert panel that reviews the diagnosis and also confirms the treatment responses. And then we use that curated information to glean clinical insights about Castleman disease and also to correlate this really extensive clinical data with each of those biospecimens. So now you don't just have a lymph node from a Castleman's patient, you have a lymph node from a Castleman's patient, and you know the entire clinical history that led up to that lymph node resection and also after that lymph node resection, what drugs were used, what worked, what didn't. It's an incredible amount of power that comes from that. And so kind of going back to that framework that I showed at the beginning of my talk, we're bringing together patients, loved ones, researchers, and also advocates through the Castleman Disease Collaborative Network, while at the same time pulling together and including the physician voice through our partnership in doing it at the University of Pennsylvania, have had a wonderful partner in Janssen Pharmaceuticals, and then also worked very closely with the European Medicines Association, really bringing together these 7 stakeholders to make really exciting progress. So what is some of the progress that we've made? Well, we've enrolled over 400 patients in our first 4 years. And for each of those patients I mentioned, we have extensive longitudinal data. So if we enroll a patient today, we get all of their historical medical records, enter those data in. We also start getting data prospectively that we enter in. So really extensive data. We get between 3,000 to 4,000 data points entered per patient, which takes days and days of medical extraction by these extractors. On average, there are about 683 clinical and imaging data elements per patient. And as I mentioned, there are about 3,000 plus other data elements that are collected on a per patient basis. And we've used this information to discover a brand new subtype of Castleman disease with really important treatment implications, a group of patients who were previously included in multicentric Castleman disease that really should be treated differently from multicentric Castleman disease. We've amazingly identified over 40 different drugs have been given off-label to Castleman's patients. There's only one drug, siltuximab, that's FDA approved, but over 40 other drugs have been used. And there's a long tail. There's a select number that have been used quite a bit. And then there's a number of drugs used just 1 or 2 times. But it's so important to collect this data. Previously, it wasn't being collected anywhere. We've used this data to enable a number of translational research studies. I've listed them on the right of the screen, but I'll just highlight a few of the things that these data and these samples have unlocked. And that's to look into the role that a pathogen, a novel pathogen may be playing, to look into germline genetic studies, somatic mutation studies, autoantibody work, pathway investigations, proteomic analyses, single-cell RNA sequencing on peripheral blood cells, morphometric analysis of lymph node tissue and bone marrow, and importantly, bringing all this together. This has led to the discovery of 2 novel therapeutic targets, really emphasizing the importance of having natural history data tied with biobank tissue and then using it. I get frustrated when I hear about people banking samples. The goal is not to bank samples. The goal is to collect samples, to use them for research so that we can help patients like myself. So let me highlight a couple of these discoveries. The first, and this is a picture of my girlfriend at the time, Caitlin, and I, around the time that I graduated from medical school, on my medical school graduation day, in fact. And then this is a picture of the two of us a few months later. In the time between these photos, we got engaged to get married, but unfortunately, I relapsed and nearly died for the fifth time battling idiopathic multicentric calcium disease. So despite the work, despite setting up registries and setting up omics studies, that progress wasn't fast enough to help me. And as I said, I nearly died for the fifth time, but thankfully multi-agent chemotherapy saved my life. But I realized that if I wanted to make it to Caitlin and I's wedding date, which was May 24th, 2014, that I would need to do everything in my power to try to identify a drug that could maybe save my life. And I'll go through this slide quickly, but just to highlight the top, these are the drugs that have been used in my case. And this is data from multiple different hospitals. I was hospitalized in a number of different places, different healthcare systems. So pulling all this data together into the registry meant that we could harmonize information on laboratory values, cytokine panels from multiple institutions. So I'm showing you data from the registry, highlighting the drugs that were used at the top, and also you can see the really intense ebbs and flows and ups and downs of key laboratory values. But the point is, a number of drugs were tried, but it wasn't until I got multi-agent chemotherapy with VDT-ACE that I had a nice year-long remission, and then another year-long remission. But here I was, fifth flare of this disease, really without options. And so what do you do when you've run out of therapeutic options? Well, in my case, I turned to the data. I turned to the data in this system to understand the longitudinal data from these multiple institutions to identify these trends. And the first thing we found was that we'd been running cytokine panels every month on my samples, and we found that VEGF, or vascular endothelial growth factor, which is a marker of angiogenesis, and also soluble IL-2 receptor, which is a marker of T-cell activation, were both very elevated in the weeks leading up to my relapse. The gray bar here is the relapse, and you can see these cytokines increasing in the weeks and months leading up to the relapse. So we thought that maybe this indicated that VEGF and soluble IL-2 receptor, or T-cell activation, could be important to calcium disease pathogenesis in my case. Now, again, if we weren't bringing together data from multiple institutions, multiple different sources, we wouldn't have picked up on this trend. Thankfully, I had stored some serum and also blood cells, so PBMCs on myself from my fifth relapse. And so I went into the lab and I began performing experiments on those samples. And I was able to confirm that, in fact, VEGF and soluble IL-2 receptor were two of the most elevated proteins when we measured 1,300 different proteins. And I was able to confirm through flow cytometry that there was a tremendous amount of T-cell activation in those samples. Importantly, the VEGF story around vascularization certainly was consistent with my clinical picture. So I had increased vascularity in my lymph node tissue. I had these cherry hemangiomas on my skin, which are blood moles that were popping up, likely related to elevated VEGF. And I had this tremendous amount of fluid accumulation, which can also be secondary to VEGF. So we wondered, is there something that's involved in all three of these phenomena that we've observed? And so we ran the proteomic data through a number of pathway analyses tools. And what we found was the mTOR pathway came up a number of times. mTOR is a really important pathway involved in cellular proliferation and communication within the immune system. So it kind of fit the bill, but we wanted to confirm, was the mTOR pathway truly elevated and was there truly increased activation? And so I ran an experiment on a number of normal lymph node samples. And so if the cells have increased mTOR activation, you're going to see that they're brown. And if they don't, they're going to be blue, counterstained. And you can see this is what a normal lymph node looks like with a normal amount of mTOR activation. And then this is my lymph node. And you're just really blown away by the amount of mTOR activation in my tissue. So we didn't know that blocking mTOR would be effective. There was no guarantee that if we blocked mTOR it would be effective. But based on these data and based on all of the work leading up to finding mTOR, we thought that if we could block the mTOR pathway with a drug like sirolimus that's been around for 30 plus years, that maybe we could control this condition. And so we began trying sirolimus on me 81.06 months ago that I've now been in remission. And I say 81.06 months because I know that I can't round up. I don't know if I'm going to make it to 82 months in remission on this treatment, but I also refuse to round down. It hasn't been 81 months. It's been 81.06 months because I'm just so thankful for every moment of remission that I've had and so grateful for all the people who helped to make this possible. The New York Times called this "Doctor Cure Thyself," which I think is a bit of an overstatement. I think it should be doctor helping himself a little bit right now and hopefully for a lot longer. But really importantly, it did enable Caitlin and I to get married. And this is a picture of us on our wedding day, and you can see my hair grew back just in time for this picture. And then a couple years ago, we had our first child, Amelia, photographed here, and this is her just a few weeks ago on her second birthday. We've gone on to show that the mTOR pathway is important in other patients, showing that it was increased and that mTOR inhibition was effective in 3 patients, going on to show that it was increased in 26 patients earlier this year, and identifying a potential candidate driver for why the mTOR pathway is increased. And this has led us to give this treatment to other patients. This picture of Joey, who was really struggling and benefited very well from sirolimus, whereas unfortunately Lisa and Sergio, who didn't respond to first, second, or third line therapy, did not benefit from sirolimus, and they push us and drive us and inspire us to continue to work harder. So this is where we were in 2012, and I'm going to superimpose where we are today. We've now raised about $1.5 million, which led to an additional $10 million in external funding. My lab now has the first NIH grant to study Castleman's. We've got over 1,200 patients in our registry. We went from there being 2 foundations to 3 at one point when we started, and now we have just one unified organization. We've won biobank. We're answering questions about the disease. We finally published diagnostic criteria and treatment guidelines, predictive biomarkers. We now have that one FDA-approved drug, siltuximab, and now there's 2 more drugs in development. The drug I'm on, sirolimus, which is undergoing a clinical trial right now, and then JAK inhibitors, which are also undergoing development as well. So in the last couple minutes, I did want to highlight that this idea of uniting to advance progress is something that has happened across medicine and across oncology in particular. The Cordoma Foundation is an example of a great group that's brought together physicians and researchers for scientific discourse. The Rare is One project that Chan Zuckerberg Initiative is leading is intended to collect and crowdsource research ideas, just like we've done for the CDCN. Count Me In is a great example of working directly with patients to get samples and medical data. The TAPER study from ASCO is a great example of systematic tracking off-label treatments. And then when COVID started, my team and I at the Center for Cytokine Storm Treatment and Laboratory launched what we call the Corona Project to systematically track treatments used against COVID-19. You can check out the link at the bottom of this page to see the database, but our goal and what we have is the world's largest database of all treatments used for COVID-19, many of which are Castleman's treatments. But amazingly, we hear about the same 5 drugs in the news every day, but there have actually been over 300 different treatments given to COVID-19 patients, and anyone and everyone can access this. We're sharing the data directly with FDA and the Cure ID app in particular, and we'll be thrilled for anyone to check it out. I don't have time to go through this in depth, but I can tell you that we've made some really important observations related to the immune response, that it's not just about being too strong of a cytokine storm, but it's about having that modulated response where you're looking for it to be right down the middle. This image is, and this is me just closing with lessons learned, is trying to show that there are about 10,000 human diseases. And unfortunately, we have treatments that are FDA approved for only about 1/4 of them. So 1/4 of human diseases have an approved compound, the other 3/4 do not. And so what I really am a big proponent of is thinking about all the drugs that are already FDA approved, like sirolimus, like ruxolitinib, like these drugs that we've identified as being candidates from our research. How many of these old drugs can be used in new ways? This is a cartoon of an old dog reading a book about new tricks. And I think we need to think about that and all these old drugs that can be used in new ways. And how can we, patients, physicians, researchers, advocates, FDA, pharmaceutical companies, how can we work together to help individual patients, to help advance for particular diseases and help to advance across diseases? And I really challenge and encourage all of you to think about ways we can bring those stakeholders together. This is my last slide, just closing with some final thoughts. The first is that I hope that my experience in my talk has shown the incredible power that comes from bringing patients to the table and actually letting patients be a part of driving forward research, crowdsourcing research, pushing forward the science, and how important this is beyond Castleman disease. The second is that hope can have many different forms. My life fundamentally changed when I went from being a hopeful person to being someone who turns my hope into action. The third is that solutions can be hiding in plain sight. The drug that I'm on was at my nearby CVS for all of those years that I was in and out of the ICU. How many other drugs are sitting at our neighborhood CVS while many of us battle deadly conditions? And I think that it should be something that we all work towards, figuring out how many of those drugs are already out there. The fourth is that we need to systematically capture data. If we hadn't been collecting data from across institutions, I would have never picked up on the trends that we saw within cytokine levels, also clinically within my case, that helped to lead us towards sirolimus. I'm sitting here today because of a drug that was already FDA approved, because of data that were already out there. They just had to be harnessed to gain these insights. And the last comment is that it really takes an army. My book's called Chasing My Cure, but I think we should have called it Chasing Our Cures, because it hasn't been me working on my own. It's really been an incredible team. And I hope all of you watching today will become a part of this team. We'll advocate for drug repurposing. We'll advocate for involving patients and the entire community in research, and help to spread the word about Chasing My Cure. I saw PDS is giving out copies of Chasing My Cure. I hope you'll read through it and help to spread the word about this journey. I wrote Chasing My Cure because I learned so much about life and living and hope and resilience from nearly dying, and it really means a lot to me to be able to share that message of hope with others. And as I shared, it has not been an effort that I've done on my own, and I do need to thank my team, many of the partners that we've worked with, and also funders for making this work possible. Thank you guys all so much for the opportunity to share my story.
David, thank you. It's such an inspiring story, and it's so motivating to see how you used hope to inspire tangible actions and real progress. It's amazing. We're all in awe of this journey, and thank you for sharing it with us. We really appreciate it.
Thanks so much, Bill, and thanks for helping to turn so many other people's hope into action through the work you guys do. It really, really means a lot.